I'm Yanzhao, an assistant professor of precision poultry management in Animal Science Department. The poultry production soared in the past century to meet the increasing demands on protein. We see that the poultry houses are much larger, and the birds are more productive nowadays as compared to decades ago. But on the other hand, the current intensive poultry production is associated with challenges regarding environment footprint, animal health, worker safety, etc., which are all under the umbrella of the overarching mission of One Health initiative. And my research is designed to address these challenges through engineering solutions. Some examples of my ongoing projects include monitoring and improving poultry production environment. I'm interested in both thermal environment, like temperature, humidity, and air environment, such as ammonia, greenhouse gases, and airborne dust and pathogens. The farm animal welfare is a big concern nowadays. Currently, animal welfare assessment is done manually by human assessors, which is subjective and time-consuming. I'm working with several colleagues and developing engineering systems that can automatically and objectively assess the poultry welfare using computer vision, computer audition, and artificial intelligence. We are exploring the possibility of using robots to encourage bird activity and to train good egg-laying behaviors. To minimize the poultry stress induced by these robots, we are optimizing the robot operational parameters like running speed, noise level, and operation frequency. My team is also trying to understand the processes of airborne transmission of poultry-related pathogens. The processes include pathogen aerosolization from the surface, transport in the air, inhalation by a healthy bird, until infection of the bird. We use modeling method to study long-distance airborne transmission among poultry farms, and we also perform on-site air sampling to study short-distance airborne transmission within a farm. I'm interested in collaborating with colleagues on airborne transmission of human and plant pathogens, like influenza virus and COVID. I'm very glad to be part of the One Health initiative team. Thank you very much.
Hi, my name is Trendl. I'm an associate professor in the Department of Microbiology. My lab studies molecular epidemiology and population genetics of microbial pathogens with a focus on the zoonotic pathogen called Toxoplasma gondii. Toxoplasma gondii is a single cell parasite that infects both humans and animals. And the parasite invade the cells, and then inside of the cells, they replicate and then can cause diseases. In human population, this parasite infect about, say, 30% of people worldwide. And in South America, this infection rate is about 50%. In human, it can cause a number of diseases. Ocular toxoplasmosis is the disease cause infection the eye and cause disease there, and it may affect the vision. And the congenital toxoplasmosis, that's the in pregnant women, when the parasite infect the person, then the parasite can pass to the fetus and cause severe disease. And in immune compromised patients such as AIDS or organ transplant, reactivation of chronic infection can cause fatal consequence in human. In animals, most animals are resistant, so they carry the parasite in a chronic state, and only a few animal species, particularly in Australia, marsupials and the New World monkeys, they are sensitive So infection can be fatal in these animals. The parasite is transmitted between cats and other animals. So cat can shed the parasite in their feces and then contaminate the environment, and other animals pick up the parasites and infect it. And then when those animals consumed by cats, the life cycle complete. In our lab, in the past, we focus on, say, molecular epidemiology and the genetic diversity of the parasite. So we analyzed thousands of samples collected worldwide, and then use genetic markers, 10 genetic markers. We can identify each samples with a relatively high resolution. So from here, you can see in Eurasia, the parasite diversity is low, and then in North America, it is higher, but in South America, there's a very high diversity of the parasite. By analyze the data we have, and we come to a conclusion that a human population expansion and migration and affect the distribution of those different genotypes. For One Health study, we are interested in develop molecular tools to detect and identify zoonotic pathogens of importance in animals and in the environment. So we'll expand what the study we did in toxoplasma into other pathogens. Thank you for listening.
Hi, my name is Dr. Marcie Souza, and I'm a professor and the director of veterinary public health in the UT College of Veterinary Medicine. I've been here for about 12 years as faculty, and I have a few research interests, most that currently focus on food safety issues. So I've been doing some research over the last few years looking at the metabolism and drug residues of various medications in backyard poultry, because these are increasingly popular in the US. And also recently had a project working with a PhD student and a few other faculty looking at public health practices, social aspects, and deception at market in the bushmeat trade in northern Uganda. In addition to research, I have a number of outreach projects, including training public health professionals as well as teachers on the international stage to improve their performance, either in things like disease outbreaks or their teaching performance. And so what do these have to do with One Health? So basically improving the performance and skills of our international health professionals improves the health of all. And then also increasing knowledge regarding food safety issues at both local and international levels. So whether that be our backyard poultry that people are keeping for eggs, or the bushmeat trade on the international level in northern Uganda.
Hi, I'm Dr. Deb Miller, and I am the interim director for the University of Tennessee One Health Initiative. I'm also the director for the Center for Wildlife Health. I'm a full professor in the Department of Forestry, Wildlife, and Fisheries and the Department of Biomedical and Diagnostic Sciences. So I'm trained as a wildlife biologist, but also a veterinarian. And more specifically, I'm a pathologist. I passionately believe that the health of every living creature is impacted by the health of every other living creature. And I believe in taking the One Health approach, which incorporates or considers that there is this interconnection. And therefore we consider all of the impacts of health on every living creature, as well as things such as healthy air, healthy water, healthy soil, which obviously is also impacted by what lives in it. So I use multidisciplinary teams to investigate the health status of wildlife populations. Currently, the largest portion of my research is looking at the pathogenesis of amphibian diseases, and specifically the chytrid fungi. I also have studies, though, looking at the impacts of elevated beach temperatures on how that impacts survival of the sea turtles that are that are incubating within those beaches during these elevated temperatures. For many years, I've also looked at the impacts of contaminants on aquatic species, sea turtles, and marine mammals. Specifically, I've looked mostly at mercury, but have also looked a little bit at other contaminants as well. And I have other studies such as looking at using assisted reproductive technologies in ocelots, and also been looking at various other wildlife morbidity and mortality events and the impacts of pathogens on wildlife populations. I believe in training. Training is so important, and I think that we can't start too young. So this is an important part of what I like to do and what I'm passionate about. I believe that my approach to my studies will impact and hopefully maintain species biodiversity, come up with new treatment options, and also could apply to human health as well, and ultimately result in a healthier planet for all.
Hello, my name is Adam Wilcox. I'm a research associate professor here in the Department for Forestry, Wildlife, and Fisheries in the Smith Center for International Sustainable Agriculture at the UT Institute of Agriculture. I've included my email contact in this slide, so you can reach out to me if you can see any potential collaborations. So before coming to the UT, most of my work was out working with conservation, big conservation, international NGOs, primarily in sub-Saharan Africa, on sustainable natural resource management, community-based conservation, and also sustainable hunting and bushmeat harvest. Here at the University of Tennessee, I work to apply social science methods and theory to improve biodiversity conservation, natural resources management, and human wildlife health. Some of my recent research that's most related to One Health is looking at perceived disease risk from human-wildlife interactions. And you'll see some of this presented by one of my PhD students, Brynn Voigh, during this One Health Day. I also look at behaviors and attitudes towards mitigating wildlife diseases. In recent times, a lot of my work with looking at wildlife diseases has focused on white nose syndrome in the US. When it comes to my disease risk work, I've most recently been looking at disease risk from bats to humans and livestock in Belize, Cambodia, and then also a bit with bushmeat in Uganda, as I mentioned with Brynn and Marcie Souza and Rick Gerhold. If you find any of this, my research interests overlap with you, please reach out with me. I'm always willing to explore new topics and learn about new issues related to One Health and wildlife, so I can apply my social science skills working with biologists and health professionals. Thank you.
Hello, I am Miksha Podar from Oak Ridge National Lab. We study microbes and microbial communities that are associated with plants, humans, animals, as well as a lot of communities from open environments, including extreme environments, high temperature, for example, environments that are contaminated with heavy metals and other compounds. And we apply a wide range of approaches from traditional cultivation to single-cell genomics, metagenomics, functional genomics, microscopy, and others. One of the major questions linked to a lot of these types of microbes is their evolution, adaptation to their environment, and in the case of microbes that are part of the communities associated with animals, with plants, with humans, is if their potential involvement in disease. We use some of the genomic and metagenomic information to either try to culture some of these microbes, as well as to essentially track them, track their abundance in the environments or in the samples that we collect them from, but also to potentially determine whether they are emerging pathogens, for example, in the case of microbes associated with animals. And I think this would be the main sort of connection to this group, to the One Health, is both developing approaches to monitor, identify such microbes, as well as to culture them and potentially understand how they can be linked to health and disease states. Thank you, and looking forward to interacting with you all.
Hello, everyone. My name is Emily Martin, and I'm an associate professor of medicine in the amyloidosis research program at the University of Tennessee Graduate School of Medicine. The primary focus of my research is amyloidosis. This is a group of diseases that initiate from a protein misfolding event, which leads to the formation of insoluble protein fibrils. As you can see here in a simple schematic, native proteins change their shape, resulting in a misfolded protein that can form protein fibrils. This is an example taken with an electron microscope of a clump of amyloid fibrils. There are more than 30 known proteins that can undergo this conversion, contributing to various forms of amyloidosis. Fibrils can deposit in all types of tissues, and with time, their accumulation, along with other molecules such as heparan sulfate proteoglycans, yields immunologically inert amyloid material that can negatively impact the function of the surrounding tissue. Early detection of amyloidosis has been an important goal in human medicine, and certain aspects of my research are focused on improving the diagnostic pathway. Additionally, our program utilizes novel amyloid-targeted peptides to develop detection and therapeutic approaches to the disease. Here you see an image of pancreatic tissue showing the deposition of amyloid as green gold material. And to the right, you see a serial section of the same tissue showing the co-localization of one of our peptides as black grains. We utilize such peptides for their amyloid binding potential. Amyloidosis can also affect domestic and wild animal species, with the most common form resulting from exacerbated and inflammatory processes that, if caught early enough, can be reversed. Prion diseases initiate from a similar protein misfolding event, but in contrast to most types of amyloidosis, prions may be transmissible. One such example of a prion disease is chronic wasting disease, which most notably affects the brain and spinal cord of deer, elk, and moose, leading to neurological impairments and behavioral changes. There is evidence that prions from chronic wasting disease can be shed into the environment from infected animals, thus contributing to its potential transmissibility. While most of my research pertaining to protein misfolding diseases studies human conditions, I am participating in the early stages of a chronic wasting disease study, along with several colleagues in the One Health initiative, that will assess the potential to detect chronic wasting disease prions in the environment and its implications on disease management. To wrap up this brief introduction to my research, it's important to know that humans and other animals can be affected by protein misfolding diseases that cause amyloid formation. Certain infectious forms, prions as I mentioned, may appear in the environment and could therefore benefit from a One Health approach to disease management and environmental safety. As a tagline, I'll just mention that early detection of protein misfolding in the clinic and the environment may impact overall health. Thank you for your time.
Hi, everyone. My name is Charles Sims. I'm at the Baker Center for Public Policy in the Department of Economics in the Haslam College of Business. A lot of what I do, I'm an economist by training, and so a lot of what I do related to One Health is around combining economic models with usually mathematical models of some sort of a measure of disease. And kind of the approach that we take is what's known as kind of bioeconomic approaches. Really two things. One is we're kind of looking at how unpredictability in the terms of disease spread influences public health responses. And so in this area, I will tend to use kind of stochastic epidemiological models, and the stochasticity comes from just an inability to predict what the disease will do in the future due to an inability maybe to predict how people will respond to a disease. And then I combine that with economic models that would kind of map things from the epidemiological models, such as deaths or infected cases, to some sort of measures in the economy, sort of job losses or GDP losses, or even the value of the mortality that comes from a disease. And I use this to figure out how risk influences the economic decisions that would be made. And so I typically treat things like lockdowns or a vaccine program as a public health investment. And then the risk tends to move things around a little bit. You're kind of speeding up some sort of public health intervention or slowing it down. You can see here kind of a picture from some work we've done on COVID, where you can kind of see the reproduction rate kind of bouncing around stochastically. And you can kind of see there what the lockdown did in kind of, the early part of the COVID-19 outbreak, and then mapping that in the bottom there to measures of an overall measure of GDP. What we found using this approach for COVID-19 was that the lockdown should have been about 1 month longer than would have been suggested by cost-benefit analysis, just because of that inability to predict what the outbreak would do. And we've used this approach in a number of other diseases as well. The other thing that I do a lot of is kind of combining epidemiological models with wildlife, populations of models of wildlife interactions with humans. What I'm doing here usually is an economic model that describes how land is being used, and then overlay on top of that some sort of spatial dynamic model of how wildlife will move in response to that land use. And then the third layer kind of in this work is usually modeling the disease spread itself. So how humans would interact with wildlife, or how wildlife would interact with livestock, and how all of those interactions sort of move the disease through a population. What you see here is a heat map kind of showing the population of elk within the greater Yellowstone ecosystem. The red areas are showing a high concentration of elk due to feeding lots. You can see without the feeding lots, the elk are much more dispersed. What we were able to do is show how these feeding lots then relate to the transmission of chronic wasting disease to livestock, and then also how that influences efforts to control brucellosis, which is another disease that's spreading through that area. Thanks a lot.
Hello, I'm Shivitoshida, a professor in the Department of Forestry, Wildlife and Fisheries, and also an adjunct professor in the Department of Microbiology. I obtained a PhD degree in pharmacy, and the thesis work was done in a public health laboratory. Since I came to the United States in 1997, my research focus has been on infectious diseases in humans and animals. So through my academic career, I have touched on the three aspects of the One Health concept: human, animals, and the environment. My current research takes a multidisciplinary approach for the control of infectious diseases, as listed in the slide, namely the development of new pathogen detection methods, a better understanding of the immunology of the diseases, and studying the epidemiology of the diseases using mathematical modeling. Many of the infectious pathogens jump around human and animals, called zoonotic pathogens. One example of zoonotic pathogens is SARS-CoV-2, the causative agent of COVID-19. Some of the zoonotic pathogens can transmit through vectors such as insects and can persist in the environment. Therefore, a multidisciplinary One Health approach is necessary for understanding and control of the infectious diseases they cause. I have worked with scientists in different fields, such as microbiology, veterinary science, food science, public health, engineering, and mathematics. I loved the multidisciplinary collaborations and have learned a lot from the experience. I also like mentoring students in the lab and classes. My major focus in mentoring is not giving out knowledge, but motivating the student in working and learning independently. If you are interested in my research, please feel free to contact me at the email address on the slide. I would love to happy to talk with you.
My name is Genita Hijab. All three names are butchered. So I'm an assistant professor at Entomology and Plant Pathology Department. I'm also One Health scholar and a Fulbright scholar to Ghana. So we do a number of projects in my lab, ranging from forest health and conservation, fungal genetics, molecular pathology and diagnostics, population biology and population genetics, and getting into microbiome and a little bit more genomic approaches. But the idea behind that is preservation of biodiversity, food security, and ways in which we can improve overall human health in general. So that is basically the sum of the projects that are done in my lab.
Hello, my name is Brynn Voigh, and I'm a professor in the Department of Animal Science. Obesity is epidemic in the United States, and it begins early in life. Approximately 1 in 7 children in the US is obese by the age of 5, placing him or her at lifelong increased risk for heart disease, diabetes, and a host of other disorders that compromise health and well-being. For very different reasons, susceptibility to obesity is a concern for broiler or meat-type chickens. Broiler chicks rapidly begin to deposit excess fat after hatch. This wastes feed, the most expensive component of production, and it also compromises welfare and the fertility of broiler breeder birds. My research uses broiler chicks as a dual-purpose model to identify mechanisms that underlie susceptibility to early life obesity. These mechanisms are important because they will allow us to then seek to identify new therapeutic targets, for example, factors in the diet that can be used to prevent that initial deposition of excess fat and to prevent obesity in children and in chicks. My research is relevant to One Health because broiler chicken in general, is a widely consumed and affordable source of complete protein that fills a critical nutrition need around the globe. Therefore, the factors that maintain the health of the environment that allows sustainable poultry production, the health of the chicken itself, are interconnected and irreversibly intertwined with human health.